Okay, today I'm going to describe to you some of our lab's work on the trade-off between reproduction and longevity. I'm going to give you details of several published studies, as well as provide some unpublished work by my co-author, Ashley Williams. To give you a broad overview, I'm going to focus on relatively few details. So if you have any questions about the studies or our study model, please don't hesitate to ask me questions during the meeting or to shoot me an email. But before I get into it, I'd like to give a shout out to the wonderful group of graduate students and postdocs in my lab, as they have completed all of the hands-on empirical work that I'm going to describe today. So huge thanks to them, including Chloe Josephson, who's one of the organizers of this symposium. So all metazoans must consume food in order to make ATP, and they use that ATP in order to as a source of energy that's going to support virtually all of their metabolic processes. So this energy is going to be used to support those processes or metabolism that's required for immediate survival. And they're also going to use that to support maintenance or repair processes that allow them to survive into the future. Now, when an animal reproduces, they will take in more energy, and the lion's share of that will go to support the reproductive effort. Now, we often make the assumption that a female can increase their allocation to reproduction by allocating less to themselves. And they do that in theory by reducing allocation to maintenance processes. And as a result, this drives what's referred to as the trade-off between reproduction and longevity, where females with higher reproductive output throughout their lives are thought to have a shorter lifespan than those that have a relatively lower reproductive output. And of course, the question is why do these trade-offs occur? So we've taken the approach of looking at processes of aging and then asking how reproduction specifically influences those processes. So there are two primary theories that are important for our research. Those are the free radical theory of aging and the mitochondrial theory of aging. By the free radical theory of aging, damage from free radicals can damage proteins, DNA, and lipids within the cell. This causes oxidative damage that drives the aging process. The alternative theory, the mitochondrial theory of aging, suggests that a decline in mitochondrial performance is what drives aging. In particular, there's multiple versions of this theory, but some think this is associated with damage to those really critical mitochondrial DNA. And indeed, these theories are not mutually exclusive, as some of those mutations will invariably come from those three free radical processes. So when we first started to look at oxidative damage and mitochondria in my lab, there was standing theory for how reactive oxygen species could ultimately drive the trade-off between reproduction and longevity. By this theory, there is an assumption that when an animal takes in more energy, they're going to produce more reactive oxygen species that will cause more damage and ultimately shorten the aging process. And I should note that we now know that an increase in damage with an increased energy expenditure is the exception rather than the rule. And so if we compare animals with high energy intake versus low energy intake, when we compare a reproductive versus non-reproductive animal, we'd have higher damage in the reproductive versus the non-reproductive. And if we compare animals with a large litter size and high energy expenditure versus a small litter size and lower energy expenditure, we would see that same pattern. So when we first started to think about this, the way researchers were addressing this question is they'd measure markers of oxidative damage. They'd also measure antioxidants that can prevent damage, and then they'd stop there. But by the mitochondrial theory of aging, it's important to realize that you should also expect to see a decline in mitochondrial performance. And at that point in time, that was a component that was really missing. So we should expect when there's more oxidative damage with higher allocation to reproduction, you'd have reduced mitochondrial performance. And so of our first study along these lines, we asked if a female would have higher oxidative damage and lower mitochondrial performance following a life of active breeding. We used wild-derived house mice as our study model. We kept them in semi-natural enclosures so that we had an environment that was more ecologically relevant. We had females that were placed in these enclosures with about 6 to 8 individuals per enclosure, and there either was or was not a male introduced into the population, giving us reproductive and non-reproductive individuals. We left them there until they were a year of age, took the males out, and let everyone come back to a non-reproductive state. And indeed, the animals that reproduced did have more oxidative damage to lipids in the liver than the animals that did not reproduce. But we expected a reduction in mitochondrial respiration, and indeed that was not the case. Instead, we saw a trend, P equals 0.06, interpret how you like, that suggested the opposite was true. So how could we have higher oxidative damage in the liver, but a reduction in the performance of their mitochondria? So when most people think about the accumulation of reactive oxygen species and the oxidative damage that's associated with that, they usually assume that there's a negative linear relationship between exposure and mitochondrial performance, or ultimately longevity. And we did too, until we started to look into the mitochondrial literature and found emerging evidence that modest levels of reactive oxygen species can actually trigger the production of more antioxidants that prevent damage, repair mechanisms to make damage go away, and they can trigger the production of more healthy mitochondria. And so this, along with the observation that in individuals that have modest exercise or modest calorie restriction have greater longevity, that led Patrick Tapia to propose this concept of mitohormesis, where modest levels of ROS exposure are beneficial, but high levels remain damaging. And so when we go back to our data, we think was happening when we saw that increase in the function of the mitochondria, we were actually in this beneficial range rather than the damaging range that most people assume when they see higher oxidative damage. So without going into any details, I want to point out that we've accumulated considerable evidence that first off, when you see oxidative damage, that is not necessarily persistent. So if you compare two different groups and one has higher oxidative damage than the other, one should not assume that that group is going to live longer because of it. We've also accumulated data that suggests with modest increases in ROS, you can get an increase in mitochondrial performance and you can get an increase in reproductive performance. So that left me thinking about how does mitohormesis fit into this relationship between reproduction and longevity? And I realized that many people in the field may interpret this graph two different ways. They may think about, okay, if we compare a bunch of different individuals that vary in their reproductive output, we would see again that higher reproductive output, shorter lifespan, but others may also think that if we follow an animal across their reproductive life, that each reproductive event would become increasingly harmful and bring that animal closer to death. But the latter really is not the case. This is a really beautiful data, older paper on inbred and outbred lab mice by Newman. And in this, he has several measures of reproductive performance across the reproductive life of the animal. So parity is the reproductive event. And what we find here is that over the first several reproductive events, a female's performance increases, and then it begins to drop. And it doesn't really reach those levels of prima parity until their 8th or 9th reproductive event, showing no loss in performance. And so how does mitohormesis fit in? So here's what I think may be going on. So this increase in performance early in life undoubtedly is in part as females gain experience, they get better at rearing their young. But I think this mitohormetic processes that improve mitochondrial performance, increase mitochondrial density, may actually help to improve reproductive performance. So the question still is, what's going on later, right? Are there delayed negative effects of this ROS exposure, or is something else going on? And so what we did here is then we dove in to look at mitochondrial DNA mutations. And it turns out there's 2 broad categories of mutations to the mitochondria. There can be damage associated with reactive oxygen species that form a specific type of mutation. It's called a transversion, where guanine and cytosine are converted to thymine and adenine. And there's lots of other types that we can cumulatively call errors during replication. Okay, so mitochondria turn over at different rates in different organs, but it can be really fast. In the liver, it turns over every 4 days. Okay, so we asked, does reproduction increase the total of mitochondrial DNA mutations, and are they more likely to come from ROS or replication error? And so we used high resolution duplex sequencing to quantify these mutations by the brilliant bioinformatics capacity of Ashley Williams. And then we looked at a few different things, and I'm going to just focus on 2 bits of data, the effective age and then the effective reproduction. So the first thing that we wanted to do is make sure that mutations accumulate with age in our wild-derived mice. So we had animals we kept in boxes. We had some middle age at 16 months and some pretty darn old animals at 28 months. And we measured mutations in their brain, liver, and skeletal muscle. And we found, yes, mutations accumulate with age in the brain and liver. Something weird's going on in the skeletal muscle. For the sake of this talk, I'm going to forget about it. Then we had animals in our semi-natural enclosures, just as I described previously, with or without a male. We put them in when they were a little bit older, at about 6 months, let them go for an additional 10 months. So they were 16 months of age when we took them out. They either did or didn't have a male in their box. And what we found, focusing on brain and liver, reproductive versus virgin mice, in the brain, there was no difference in total mutations. There was no difference in those that were associated with ROS, which means there was no difference in what was left. When we looked at the liver, there was something really interesting going on. So the number of ROS-mediated mutations was higher in females that reproduce, and interestingly lower in those that there was a lower replication error. But when we looked at total mutations, they weren't different between groups. And I should note that just those variables in red are the ones that were statistically significant. So I should note that we also compared the basal and maximum respiratory performance of the mitochondria between these 2 groups, virgin versus reproduction in the liver, and they were not different, saying there was no functional effect of that difference in mutation. The other thing I want to point out is that females that reproduce were more than 2 times more likely to die during the study than the virgin mice. So that tells us there was a negative effect of reproduction on longevity of the females. And I have some ideas of why that's the case, and I'm happy to discuss that with you further. But I think most interestingly, we then took the females that reproduced and looked at their cumulative mass of their pups at weaning as a measure of investment, and found that those females that invested the most in reproduction had the fewest number of mutations in their brain and in their liver. So my take-home message is despite the high energetic demands of reproduction, our data provide little evidence that bioenergetics really is what's playing a role in that trade-off between reproduction and longevity. Instead, the process of mitohormesis may protect or even improve reproductive performance. So thanks to our funding sources and all of those who have helped our lab group out.